We remember that the ribosomes are the locations, the spots of protein synthesis. And we need to know the locations or the spots of the catalytic activity. And your book details this, the 3 different spots that you're responsible for knowing. There's the P site, the A site, and the E site. The way I remember this is I always think of the P site as being the processing position. The A is the addition section, and the E is the exit section. So if you're looking at a ribosome, you'll see that they all have this basic outline of where your shapes are. And it would work out in such a way that on your ribosome, and I think it would be best if I just showed you a picture rather than anything else. So let me show you a picture. So there's your ribosome, made of the small and the large subunit, and there's E, P, and A sites in that order. So what the E site is, or what the P site is going to do, is it is the processing site. It is the spot that holds the tRNA. That is carrying the polypeptide, or protein, we could say, chain. So carrying the polypeptide chain. There's the A site, which is going to be the location of the next amino acid, the next tRNA molecule. So this is where the next tRNA is at. And the E site is going to be the exit point of the empty tRNA. Mention is that you can have multiple ribosomes. I'm almost done. That are reading the same piece of mRNA. That means that one single piece of mRNA, one transcript, can serve the purpose of producing many proteins that are the exact same, of course, because it's the same transcript, at one time. So you can use many ribosomes reading the same piece of mRNA at one point in time. So in other words, going back to the picture that I just had up, you of course can agree that the red is the mRNA sequence there, right? You could have a ribosome that's maybe 40 bases this direction, reading the mRNA as well.
Would it reread it when it keeps going through the next one?
That means as you have this polyribosome setup, that's what it's called, you can produce lots of the same protein because you had many ribosomes reading the same piece of paper. It's like setting a copy of my notes right here and having 5 students come directly around me and read the notes. I mean, there's nothing stopping this transcript being translated into knowledge by more than one individual. Right? We could share this piece of paper. So ribosomes can share the transcript. Right?
Are like the ribosomes set to make one protein, or can they make a bunch of different?
Once they've read this mRNA, they could reread it again. Absolutely.
Can they make it different?
You can't because this transcript is limited to one thing. Just like this one piece of paper.
Ribosomes read different.
Oh, absolutely. They could read a totally different transcript the next minute. It's like if you were the ribosome, you have to. If this is the only paper in front of you, if this is the only mRNA, then this is what you're reading. But if there's some other transcript that shows up, there's nothing stopping you from reading it. Cool. The last little bit in this chapter, in this unit, is going to be over some mutations. So let's talk about a mutation. What is a mutation? A change? Okay. We saw a while ago that you could actually have a mutation that didn't matter at all because of Wobble effect, right? So mutations. Now this is an event in which there are changes in nucleotide sequences in an individual's DNA. Now, someone had mentioned in first period, they said, well, why can't we say it's base changes? I guess it is. The only difference between nucleotides that are DNA nucleotides would be the fact that their bases are different. Nucleotides have sugar, phosphate, base. So you could say different bases or different nucleotides. It's the same thing. And there's a specific type of mutation that I want to talk about, which is a point mutation. This would be a mutation at a single nucleotide. Or, if you wanted to, you could say base, because it is the incorrect base. That's the only difference between the nucleotides is their bases. So, it's a bad nucleotide, which means it's a bad base. Good. And I want to talk about two broad categories of point mutations. So we could divide this up into two categories. We've got our base pair substitutions. Or we could have the base pair insertion or deletions. So different types of point mutations. But in either which case, we're going to be changing just one nucleotide. There's one mistake. As far as base pair substitutions, there's a couple of different types. The first one we can make mention of is the silent type. We talked about a silent mutation a while ago. That's one in which there is no difference in the overall protein that's produced. This could be because the mutation is in an intron.
In a what?
Intron. Do you remember what an intron is? Intron was a non-coding section of the DNA. It was cut out. Remember, the exons stay, the introns go. So the introns are the pieces of mRNA that are non-coding, at least in that specific protein. If you make an error, a mutation in an intron, which doesn't end up in your final mRNA piece, well, then it doesn't really matter. Right? And then also, we saw a while ago that another reason was because of the wobble effect. So there are two reasons that your substitutions could be silent. They occur in an area that doesn't matter, or they occur at the third position where there's some flexibility. Right? There's also two other types of base pair substitutions. I want to go over missense mutations. In a missense mutation, we've got the wrong amino acid at the wrong time. So this causes the wrong amino acid to be added. Missense mutations are usually bad, to be honest with you. However, then sometimes they could be good, because if you put a wrong amino acid in a spot, but the amino acid is pretty close to the same, it may not have an effect overall. And usually, if cells are producing the wrong protein, because they've got a missense mutation, they usually recognize that. So they kill themselves. It's true. Every once in a while, you could have a nonsense mutation, and that's one in which... Travis, did you have a question?
No.
In a nonsense mutation, the mutation causes a stop codon to be added. So the mutation changes the codon to have a premature stop codon. What that means is that the protein is being formed, or you're transcribing your mRNA, and then it stops all of a sudden. The cell recognizes again that it's produced the wrong protein, or it's just neglecting to produce the protein at all. Usually,
Like a non-finished protein.
this would be a protein like you started building and then you stopped because the directions just were randomly cut off, and you kind of recognize, well, obviously I can't use these directions, so you throw them away. It's kind of what the cell does in this case. But we've got your this again is like when you took, you know, a sequence of like CAT and you made it CTT. You changed one base, a single base pair substitution. Could make no difference. Could cause a bad protein. Could cause a protein that just really isn't even formed because it stops. These are usually much worse though, the insertions or deletions, because regardless of if it's an insertion or deletion, this is certainly going to result in a messed up and off reading frame. Once you add or delete a nucleotide, your codon sequence is going to be completely off. The example that I always use to help people understand is that this would be just like a scantron and messing up on a scantron. Think about it. Let's say that you were on question 2 and you accidentally answered question 2 on both the 2 and 3 spot. And if it was a 50 question test, that means that your last answer would be at spot 51. You realize that if you put that answer in twice, you've messed up everything from that point on. Or what if you didn't answer a question? What if you answered question 3 in the position of 2? Well, then everything from that point is messed up as well, right? That's analogous to your reading frame. These are disastrous as well. It results in the protein just not being produced. Now again, in almost any case, if you're messing up the protein, the cell is going to recognize that and probably shut itself down. But every once in a while it doesn't. Those usually are cases like cancer or some other abnormality, autoimmune diseases and such. Cool? Let me show you an example of this, of how this works out in application. I'm going to show you a codon or a 3 nucleotide sequence in DNA in 2 forms. Let's say that an individual's DNA read at 3 nucleotides, it read CTT, but it was changed so that it was CAT. This would be the wild type form, the normal, and this would be the mutant form of the gene. Would this be an insertion or deletion? Would it appear? Would it be like a substitution? Yeah, it seems like a substitution. Now this may be silent, or it could be missense, or it could be nonsense, but it depends what happens. And let me show you what happens when you create this mRNA. We would expect the mRNA for this first one to be what? GAA. And this one would be